Hi. Thank you all for coming to watch us give our presentations. My name is Abigail Dommer, and I'm a postdoc at UC San Diego. And it is an honor to be here as part of this year's T12 cohort of really amazing scientists. And I'm excited to talk to you today about my research and my journey into chemistry, really. So I grew up in a suburb of Columbus, Ohio, and I never really thought I would be a scientist, and certainly never thought I'd be a chemist. And my mom likes to tell people that as a kid, the only thing I wanted to be when I grew up was famous. So, that's not super helpful in terms of deciding on a career path. But on the outside, it seemed like I was setting myself up to go to art school. CCAD, the Columbus College of Art and Design, had these Saturday morning art classes. And so every year from first grade to high school, I attended these art classes. And so, when it came to choosing a college, art school seemed to be a natural choice, but I wasn't completely sold on the whole art as a career thing. And I wanted the freedom to explore other options. So I ended up choosing Washington University in St. Louis. And I know our next speaker actually went to WashU for their PhD. So it's kind of like a WashU fest over here. So WashU is this beautiful private liberal arts school in the Midwest. And I decided by then that I wanted to pursue fine arts, but I wanted to get a biology minor in addition to that. Because I thought maybe I'm well suited for some kind of career in biomedical illustration. It seemed to have the intellectual rigor that I valued, as well as the technical aspect of the art that I also loved. And it seemed like the perfect combination of my skill sets. So my first year at WashU was really defined by these two professors, Professor Mary Borgman, who taught my Drawing 1 class, and Professor Loomis, who taught Gen Chem 1. So Mary Borgman is this just phenomenally talented portrait artist who taught my drawing class and completely revolutionized my personal art practice. But why am I telling you this? Because we're at a chemistry symposium. Dr. Loomis was the one who dragged me away from the art school. So he took me to the dark side. And I guarantee you he has no idea who I am. But his class just completely rewired my brain. And if you know Professor Loomis, you know that he's just this incredibly loud and charismatic and inspiring person. And, straight away in Gen Chem, we were learning about the atomic orbitals of hydrogen and plotting them in spherical coordinates, using the solutions to the Schrodinger equation. And I just thought it was so beautiful, the way that the math worked out and the way that it can be visualized. And so, before I knew it, I was just abandoning my whole art and biology thing to be a chemistry major. I think he'd be proud of me. I'm not sure, though. I got to be in his class. And, people always think that moving from art to science is such a big switch, but I think they're actually not so different. At the core, right, you're really just trying to interpret complex information, and you're translating observables that the world is showing you into human interpretable language. So whether it's, trying to convey feeling or time of day in a painting, or trying to decipher a data set that you get out of your spectrometer, either way, you're being creative and you're solving problems. And to me, these skill sets are very similar. So it wasn't much of a leap for me. And just as an aside, I definitely did not abandon art at all. I still have a personal practice. And I'm also illustrating journal covers for my own publications, and that's something I really enjoy. And art still definitely follows me around in my work today, and you'll see some of that later. So there are many people who directly impacted my experience at WashU and inspired me to go into research, but I'm going to focus on the two who actually contributed to my decision to go into research. And so I ended up joining Dr. Blankenship's lab, and he worked on photosynthetic bacteria. And, you know, I was terrified to join a lab. I didn't grow up with any scientists around me. So I had no idea what this was about. I thought maybe I'd have to go to the PI and like have a fully formed research project idea ready, but turns out that's not the case. And so Dr. Blankenship set me up with Erica, who was a grad student at the time and is now faculty at the University of Wisconsin-Madison. Congratulations to her. So proud of her. And she made the process so easy for me. She set me up with a project, and she was really the one who encouraged me and inspired me to go to grad school. And I'm so grateful for her, for all of the work that she put in. So after all of this, of course, I ended up at UC San Diego pursuing a chemistry PhD. I thought initially I wanted to work with materials and solar energy, but as soon as I met this individual here, Kim Prather, and learned about her work, that was it. I was sold. No other research was going to do it for me. So Kim Prather is the director of CASE, which is the Center for Aerosol Impacts on Chemistry and the Environment. And their whole thing is to study Seaspray aerosols and how Seaspray impacts climate. So I'm going to, maybe I won't skip over this. So it turns out you need an aerosol in the environment to form a cloud. And clouds actually play a huge and not well understood role in regulating the global energy balance. And Seaspray in particular, because the ocean covers over 70% of the planet, and because the ocean is full of these complex chemicals, it's very difficult to study. But CASE has been able to literally bring the ocean into the laboratory. And I have a picture of that here. They siphon ocean water into this giant tank, and then they can use a paddle to create waves, and then they can collect the sea spray off the surface of the waves as it's coming off, and then study it. And they can study how, you know, how sunlight interacts with each particle. They can study the size. They can study all kinds of things, how a cloud's going to form on it, how ice is going to form on it. And this will ultimately help us better understand how aerosols themselves impact climate. And so one of the things that, you know, I was just fascinated by the fact that Seaspray aerosols aren't just salt water. It's, you know, the ocean is just this giant bath of chemicals, really. You have this biochemical engine made up of bacteria and enzymes, and you've got all of that plant life and animal life and viruses, all of this like gunk that settles to the surface, kind of like oil and water settle in layers. And then when a wave breaks, it launches all of that material into the atmosphere. And so you actually get living viruses, living bacteria, all of this stuff comes out, and it's not just salt water. And so this complexity makes it even more difficult to predict how these properties are going to influence climate. So I'm not going to talk about this, but I did take 2 years off to teach. You know, I think a lot of people when they go to grad school have this moment of, is this really the right thing for me? And most people don't act on it, but I definitely acted on it. I was like, peace out. I'm going to go teach for a while. And so I ended up teaching middle school and high school, and then I ended up teaching at community college, and it was a wonderful experience. And one of my mentors at UCSD, Judy Kim, actually was the one who convinced me to come back. She was like, if you want to teach, you need to get your PhD, you need to come back. And that was it. So I ended up going a different route. I still wanted to study Seaspray because that was the research for me. It still is. I love aerosols. I love aerosol chemistry. But I ended up working for Romy Amaro. And Romy Amaro is a computational chemist, and her group largely focuses on, you know, rational drug design and protein design and the biomedical stuff. But I, along with a postdoc at the time, Jamie Schiffer, sort of launched her group into the CASE effort. And what we contributed was we started we were able to build models of Seaspray aerosols in their full chemical complexity. And at this point, you know, people really didn't know. People didn't know what these things looked like. People still don't really know what these things look like in their natural state once they're ejected from the water. And so we were actually able, and by we, I was actually able to model these things. And so what you can see here are, so first of all, this is a 3 million atom system. And if you're familiar with computational chemistry and molecular dynamics, this is on the large end of systems. This is like a very, very big particle, lots of atoms. And the two things I want you to notice here is the blue is water, and the orange wiggly guys, those are lipids. And you don't really expect lipids to come out of the ocean, but lipids come out of the ocean, and they tend to separate just like oil and water, except they separate in weird ways that you can't really predict. And this is interesting because we would expect the lipids to fully cover the surface, but what you end up finding is that the lipids form like patches, okay? And in the regions where the lipids meet, you get water evaporation and things like this. So this has deep impacts for the chemistry of our environment and water absorption and cloud formation and things like this. So then when you add complexity, it makes the surface even more exposed and even more likely to take on water. So all of these findings are just things no one has ever seen before. So this paper is in progress. No one's stealing my work. Trying to get it out in the next couple of weeks. Okay. So just to give you all whiplash, I think I'm running out of time, but I'm not sure. When the COVID pandemic hit, it turns out that modeling giant lipid balls really comes in handy when you're trying to model a full coronavirus. 2 minutes. Thank you, Bethany. So when the pandemic hit, Romy was like, hey, should we maybe try to model the coronavirus? And of course, I was like, yes. I'm sort of like now the lipid modeler of the group, and was totally into that. And so I and a couple of postdocs in the group, and now a couple of other grad students in the group, we all kind of came together in this big collaborative project to build the coronavirus in its full atomic complexity, and then run it, and then simulate it with all atom molecular dynamics. And so this model had, 300 million atoms in it, including the water box. And so this is just like astounding, okay? This is like really pushing the limits of high performance computing. And we ended up with this model winning the 2020 Gordon Bell Special Prize for COVID-19 research, which was very, very cool. It's just like, career defining win for us. We're all very proud of this work. But then, of course, 2020, it's not good enough. We have to keep going. And so we were able to actually combine my aerosol expertise with the coronavirus work, because now we're thinking about respiratory aerosols, and the fact that the coronavirus is airborne, right? And so we pushed this project even further. We were able to model in full atomic complexity the coronavirus inside a respiratory aerosol in full atomic complexity. And this is something no one has ever done before, okay? We had to figure out what's in a respiratory aerosol. We had to go to the experts. We figured out how to model mucins. We figured out how to model just all of this gunk inside of a respiratory aerosol. And this is going to help us figure out, how does the virus stay viable in the air when it's being transported, right? Because the virus, when it touches atmosphere, is going to immediately die. What is keeping it protected? And we think it's actually, mucus actually plays a significant role in this process. And so I forgot to mention, this model that we built is 1 billion atoms. And this is, one of the largest models that's ever been built with full atom complexity and actually run successfully using molecular dynamics. And this was a finalist for the 2021 Gordon Bell Special Prize. And so moving forward, and wrapping up, I know I'm like running out of time here, but I mentioned before that aerosols that come out of the ocean can contain viruses, and those viruses can stay alive. And so moving forward, we want to understand, aerosols as environmental pathogen carriers, right? Because if you think about it, it's not just humans that live on this planet. We're not just spreading disease from one human to another human. These respiratory diseases, they're interspecies diseases. There are plant viruses and bacteria that can get spread in the wind. And these things can impact food scarcity and climate change and human health and biodiversity loss. And so moving forward, this is sort of where we're going to go. How do aerosols really play a role in the changing global climate, especially with global warming and so forth? So with that, I want to thank Chemical and Engineering News for hosting us today. And I want to thank Laura Howes, who was amazing. She was the journalist who wrote my article, and it was just so lovely. I want to thank Romy Amaro and Kim Prather and Vicki Gracian, who have been just wonderful mentors to me. All of the NSF case members and our Gordon Bell teams. And here's a picture of me with our group and my family and my 3 cats. Yeah, so thank you. Happy to take questions. Sorry I went over time. Pretty sure I went over time, but thank you.
Thank you so much, Abby. I really enjoyed your talk. We have a question in from Colin Durkin, who asks, is there the potential for the aerosols exiting a whale's blowhole to form clouds?
Yes. I would say so. I don't know what chemicals there are in a whale's, in the aerosols that come out of that, but if they get suspended in the atmosphere, it's possible. We need to run that through a cloud nucleation machine.
Let me ask you a question that I have. I know there's so much artistic work that goes into your research, and you spoke a lot about your work with art at the beginning of your talk. And I'm just wondering, is there a project in science where you think like more art or a more artistic depiction would be helpful in engaging the public?
Oh my gosh, absolutely. I know that, it's like one of those things, like the public just doesn't trust scientists. And I think that, and it's actually one of my goals. I want to write and illustrate a children's book that makes science beautiful and makes it inspiring to kids. And I think that kind of thinking will really work on adults too. You wanna be able to translate this complex, complicated, scary science into something that your audience can appreciate. And especially since, we're learning that logic doesn't necessarily help influence people. It's getting at your emotions that will make people change their minds. And so I kind of think that art can play an important role in that. And it can even be, it doesn't have to be like visual art, and it can be music and, any other kind of dance, any other kind of art that gets people thinking about science and trusting scientists again. Yeah, great question. Thank you.
All right. Thank you so much, Abby.